So we're moving into chapter 3, which is on how biologists organize the biological world, how they classify different things, also called taxonomy, but we're going to stick with classification. And it comes down to the same way that science kind of depends on our need to explain why things work or how things work. This comes from our absolute need to both put names to things, label things, and to understand how things are related to one another. Is that labeling things is important in us because we have language, and since we're a structured society where a lot of the structure depends on families and interfamily relationships, we like to know how things are related to one another. This, incidentally, these guys didn't live with these guys, but what the heck. If you can't read the caption, it says, now this end is called the thagomizer, after the late Thag Simmons. Labeling. We gotta label stuff. What's it called? We'll call it, took out thag, so we'll call it the thagomizer. So, chapter 3, I don't always remember to label these. I do try to say it at the beginning. Just realized if I'm starting a new chapter, I should put that back up there. I don't need to leave it very long since you can freeze frame it if you still need access to the times that I'm around. So what makes this thing related to that thing? This thing more closely related to that thing, this thing more distantly related to that thing. How do we find the relatedness of one group to another? Well, if you go back in time, the ancients very often looked at you can see this in how they structured the first chapter of the Old Testament, Genesis. How were things created? Well, they lumped the groups together, and on one day, everything that flew was created because that's what they did. Analogy is linked by function, by what things do. And so to the ancients, birds and bats and butterflies all belong to the same group because they're all analogous. They all did the same thing. And things that swam in the water, things that were long and skinny. In the Bible, there's a lot of long, skinny, wiggly things that translate always as serpents because they were all considered to be one type of serpent or another, even though one of the descriptions very obviously is describing a type of roundworm parasite that crawls under your skin, makes you feel like you're on fire. Fiery serpents. But they didn't know. Serpents, eels, whales, fish, they put them together by function. And that's a little bit misleading. A dolphin, a tuna. One's a mammal, one's a fish. An ichthyosaur looked an awful lot like a dolphin. That was a reptile. Why? Why do you have these different things that resemble one another and do? Because, well, it is because they're doing the same thing. If you're going to move through the water quickly and catch things to eat, then there's a certain shape and a certain streamlining that you need. If you're going to be in the air, you probably got to have wings. So what you find is convergent evolution, where you'll find things that look similar to one another because their function requires a certain form. is that long and skinny turns out to be a great form for burrowing and getting under things and in tight places. So that long, skinny form shows up in all sorts of groups that aren't really related to one another. Snakes and roundworms and eels, not closely related to one another. But you get convergent evolution where they look superficially like each other because they're doing the same stuff. Is that to get into tight places, a long, skinny shape is very, very useful. So you find it in a lot of places. So it turns out analogy is not that great for determining relatedness. Homology or homologous features, I'm not going to write that out, but feel free to do it yourself. So you look at a bird and a bat. Even if you start poking inside, is that a lot of the bones very similar to one another, but when you get into the wings, then the wings are built very, very differently. When you look at the outside, is that bats are covered with hair, birds are covered with feathers. Birds use those feathers as a flight surface, is that they open their wings and it's all feathers. Bats basically have these long finger bones, and there's webbing, like here, between the finger bones that's what they fly with. If you try to open up a butterfly, good luck. Their skeleton's on the outside, is that they're essentially hollow and their organs are on the inside. So their wings are not really built anything like a bat or a bird's wings. But a bat's finger bones are actually more similar to our finger bones than they are to a bird's. A bat's hair is more similar to our hair than they are to a bird's. So when you start to look at how things are built, you start to realize bats belong in a group with us, birds belong in a separate group with things that have feathers. And as they looked at the inner forms and found evidence from the fossils that a lot of birdie type dinosaurs that didn't fly, but had a lot of bird-like structures to them, but also had feathers, that probably the birds are an offshoot, a descendant of those particular types of dinosaurs. You're looking at structure. And while I'm on the subject, I'll talk a little bit about fossils. But before I do that, there's the other type of evolution. Bat ancestors were more similar to shrews, which are kind of mouse-like, than they are to birds. And shrews are still around. And was a group that stayed, working the ground and eating bugs that stayed the shrews. And then there was a group that first was probably gliding from tree to tree, and then better and better control is a good thing. Is that you see selection of more successful flappers and flyers, and eventually a system that's legitimately wings. So the shrews and the bats are related to one another, but they're not very much like each other anymore. They're divergent. Is that a whale's closest land relative is a hippopotamus. Not that similar anymore. Is that hippopotami are pretty good in the water, but they come up on land to browse and eat. Is that whales' ancestors got much more linked into the water all the time. Is that the natural selection there was to select more efficient swimming structures, and also for sexual selection, a type of reproduction that didn't require getting up and vulnerable on the land. But you get divergence. Hippos don't look anything like whales, but their ancestry led to the two divergent groups. God, I can't spell. There we go. Fossilization, fossils. To some extent, this is connected to fossils, which they've been digging out of the ground for thousands of years. So you get these things, what are you looking at? You're looking at bones, you're looking at shells, you're looking at teeth, hard parts of animals. Is that your typical fossil is something that died, and then before it can be completely recycled by the things that eat it or the things that decompose it, it gets hidden, it gets isolated, it gets covered usually in some sort of material. The most likely material that produces fossils is sediment in bodies of water. So we don't have certain great fossil records of a lot of dinosaurs. Dinosaurs were land animals, and most of the fossils involve dinosaurs that were land animals, but they, once upon their death, they wound up at the bottom of a big lake, or they wound up on the bottom of the near ocean environment, or the giant sea that was in the middle of North America, way back when the dinosaurs were roaming around. And things got down there and they pulled the flesh off, and sometimes they pulled them apart, eating them, but they didn't eat the bones, and they couldn't really decompose the bones. They couldn't eat the shells. They couldn't decompose the teeth. So typically, fossils are the remnants of hard parts. To get fossils of things that were soft, you needed a very particular type of covering. Usually the sediments on the bottom of a shallow body of water where there was almost no oxygen in the water. Really limits the number of things that could get to the bottom where something had dropped and chew it up. Plant fossils come from those types of sediments. And if you know anything about swamps, you know that those sediments at the bottom of that thin, smelly, swampy water is very smooth and slimy kind of sediment. So it not only preserves soft parts like plants, we have some plant fossils in the lab you can look at that were made that way, but also imprints. Is that some of the earliest birds that we know of fell into that type of water, sank to the bottom, didn't really get pulled apart because there was nothing big enough in there to pull it apart. And the slimy, thin sediment actually molded around their feathers. And sometimes if you crack the shell apart just right, you can actually see the imprints of feathers around the bones of those things. So, but for the most part, you're comparing skeletal elements. You're comparing teeth. Sharks don't fossilize real well because they don't have a hard skeleton, but they got pretty hard teeth. And a lot of our fossil record on sharks comes from their teeth. Things that are crispy critters, like big crabs and stuff, we've got a pretty good fossil record for it. Things that are really tiny and have an outer exoskeleton tend to die and then get eaten by scavengers, so we don't have a great record on insects and spiders and that sort of thing. Or on bats, for that matter, because bat bones are pretty thin, and for the most part, they can be pretty well consumed by things that are chowing down on the dead stuff dropping to the bottom of a body of water. You can get coverings of sand, you can get coverings of volcanic ash, you can get coverings of plant sap, that's where amber comes from, but most of the time you're looking at bones, and the idea of we can use these bones to figure out what relatives these animals had led to dinosaurs being recognized as reptiles. I don't know that they still would be if they were still with us today. And it's homology. You're looking at comparisons of internal structures based on the structures that you know and the structures in the fossils. And from there it went to comparing living things the same way. Eventually, it was even applied to early structures. How does an animal develop from the very first cell until it's actually ready to get out and get around in the world? It's an embryo, eventually a fetus. And there was a scientist named Ernst Haeckel. There's a link in the book about Haeckel. And he developed a technique where he could expose a developing egg, get the shell off of the membrane inside without breaking the membrane. And he could actually watch development happen, and drew all sorts of pictures, and started to see structures that when he looked at fish, when he looked at birds, when he looked at mammals, when he looked at reptiles, he'd see a lot of very similar structures in things that, once they came out of the egg, didn't really look anything alike. Bird, lizard, you know, pig that doesn't even have an eggshell. Those are a little trickier to do the development of, but you know, you can do it. And he thought he saw patterns that turns out what he thought he saw wasn't right. And I gotta talk about this. Haeckel did this idea of, gotta remember this a long time ago. People liked really big words. There was a point when you were an embryo and you had gill slits here. You had loops of arteries, like the loops that would be in gills. You had a tail. You had more, you had basically a paddle. Your fingers were all webbed. Haeckel looked at these things. You actually went through a stage where your kidney was very fish structured, and then reptilian structured, and then eventually mammal structured. And all sorts of different relatives of ours have those same gill slits and the same tail and the other sorts of things. Now, we now use that to go to find connections in things that you don't necessarily see the connections of once they're up and about in the world, because, I'll talk about that in a minute. This, Haeckel actually thought that he was watching the embryos relive their distant family history, that the development of the embryo reconstituted, recapitulated, replayed their family history, their evolutionary history. That there was a point in all of these embryos when they were actually fish, and there was a point where they were actually reptiles, and then if they were birds, they went on to be birds, if they were mammals, they went on to be mammals. Which turns out not to be right. Yeah, we got gill slits, but we never, you and I never had functional gills. We never were legitimately fish. We never were legitimately reptiles. We just share features during development. Evolution is driven by conditions. Developing embryos are in very well-protected conditions. So they tend to retain a lot of features across a really broad range of different embryos. And also, any change, any mutational change that changes development really early is gonna change stuff down the road quite dramatically. So you don't see a lot of shifts in development unless they're really, really useful. So it's, Haeckel pretty quickly was refuted in terms of how the embryo is working. I just realized I've got this overhead that shows, you look at, and I got it backwards, these two bones in your arm are also in the legs, the foreleg, the front leg of a lizard. It's there in a bat, although the second one's very much reduced and not attached to the same muscle. That's what, a vestigial bone isn't doing the major function that it does in relatives. It doesn't mean it doesn't do anything. Some people think it does, means that. But it's kind of got something else that it's doing. A lot of times more minor. Is it the bones in a pterosaur, the things that flew around during the dinosaur area? There it is in a bird. But you can see the finger bones are very modified here. In a horse, in a plesiosaur, the big paddle guys with the long necks that lived at the age of dinosaurs. And down at the bottom is a dolphin. Same bones, a lot of the same bones, similar structure. So it tells us who's related and not related. And looking at embryos is also very useful for that. We now do something else. In a couple of chapters, we'll get into more detail, but we've already talked about how we have these codes, these DNA codes, and the codes produce proteins. Well, the DNA is a long sequence that can be translated into the long sequence of amino acids that's a protein. And when they couldn't really analyze DNA sequences, they pulled proteins apart and analyzed those sequences and used them to compare. is proteins are, structures. DNA is structure. And an awful lot of what we do now compares the DNA sequences from one potential relative to another. In fact, they use something called a molecular clock. Your most common DNA mutation is a change in one little bit on the long sequence. They call them point mutations. We'll talk about that later, and you'll need to know the term then. So here's an ancestral population, and here's a descendant population, and here's another descendant population. So they split from one another at some point in the past. This starting population was genetically pretty similar. One branch accumulates point mutations in places that aren't important. You got a lot of DNA that's not doing anything really, really critical, so it can mutate at a point and it virtually never screws anything up. Mostly those neutral mutations I talked about. The other branch is accumulating its own unique point mutations at about the same rate. So, and I'm making these numbers up. Say on average you get a point mutation along each branch once every 5 generations. So you look at a stretch for this population and you compare it to a stretch from this population, and you find 20 point mutations that don't match. So it's 100 generations since they split off from that common ancestor. Molecular clock, point mutations act like ticks in a clock going at a rate that we're not absolutely sure is reliable or steady. There's iffy aspects of the molecular clocks, but this is how they decide who's related to whom and how closely they're related to whom, mostly today. Does this belong in the same species? Is it a subspecies? Is it a separate species? They look at a lot of DNA differences. Okay, that's where we're going to stop on this part of the chapter.